Assalamualaikum, hope you all are fine. So, in today's lecture, we will discuss what are the stem cells and what are their basic properties. Similarly, we will also look into the tissue specific nature of the stem cells, what are adult stem cells, and for the adult stem cells we will be considering an example of the hematopoietic stem cell, because these stem cells play a very important role for the development of all the variety of the cells of our immune system. And similarly, we will also see how the age and environment and the genetics basically affect the production of the stem cells, because the stem cells can give rise to the variety of the cells in your body, but the lineage commitment of these cells is greatly impacted by the genetics and the various other factors such as environment, various types of the mutations as well. So we will also look briefly into these factors as well. And similarly, in this lecture we will be discussing in detail the hematopoietic stem cell, because it is directly related to the immunology and the immune system, and we will see what are the different cells lineage commitments which is done or which is undergone by the hematopoietic stem cells for the production of the variety of the cell lines of the immune system. Okay, so generally, what are actually the stem cells? Stem cells are those cells of your body which have actually ability to give rise to any type of the cell for your body. Okay, any type of the cell. It means that a stem cell basically have the ability to differentiate into the variety of the cells. And the first type of the stem cells which you encounter is the embryonic stem cells. When you are at the embryo level, the embryonic stem cells are present in your embryo, and these embryonic stem cells basically have the ability to produce diverse types of the cells, whether it's related to your heart, your kidney, your liver, your digestive system, your nervous system. Every type of the cell which is required for building these tissues and organ systems, all the types of the cells can be formed by the repeated divisions of these stem cells. Okay, so these that's why these stem cells at the embryonic stage are called as pluripotent stem cells. Okay, so first of all, you have to consider this thing, that the pluripotent stem cells, what are actually these stem cells? These stem cells basically have the ability to produce diverse variety of cells. Different types of the cells can be produced from them. Any type category of the cell from your body can be produced from the stem cells, and that's where they're called as pluripotent, except the placenta cells. Okay, so placenta cells cannot be produced at this level, but the all other types of the cells are produced from these pluripotent stem cells. Okay, so in general, the stem cells have the two capacities, they have the two abilities. Okay, they not only self renew mean they have the ability to reproduce themselves, they have the ability to produce their own kinds. They reproduce, and they produce definitely the daughter cells, and all these daughter cells basically have the characteristics of the stem cells. They have all the surface markers, all the genetic makeup, and all the properties which are identical to their parent stem cells. Okay, so they can repopulate themselves. And second important feature of these stem cells is that they can differentiate into variety of cells. Okay, so diverse types of the cells can be produced from these cells. So these are the two important properties. And when these stem cells are present at the embryonic level, they are called as pluripotent, because they can give rise to any type of the cell related to any organ, any tissue of your body. So they are called as a pluripotent, except the placenta cells. Now the what basically happens actually when these stem cells basically produce all the different parts of your body, whether it is a brain, it's a bone marrow, it's a lung, liver. When all the organs of the organism basically has been developed, now the stem cells which are present in these organs are highly specific to their location. Okay, so now these are called as adult stem cells. Why the stem cells which are present in an organ are now called as adult stem cells? Because these stem cells, now whenever they are undergoing the differentiation, they will produce the cells which are highly specific to any particular tissue or organ in which they are present. Okay, for example, let's suppose a stem cell is present in the liver. Okay, so every time that stem cell is going to divide, it is going to produce the hepatic cells, that is the liver cells. Okay, and which are very specific to that particular tissue or organ in which it is present. Similarly, when the stem cells are present in the other parts of the body, they are going to produce the tissue specific cell types. Okay, which are specific to a particular tissue, or in generally it is specific to an organ or organ system. Now they don't have the ability to produce any cell of your body. For example, you have a stem cell in the kidney, and it doesn't have the ability to produce the cells of your liver. Okay, to produce the cells of your lung or your brain. Okay, only the stem cells which are present in your liver will be able to produce a stem cell of the daughter cells related to the liver. Okay, and similarly the cells which are stem cells present in your brain will be developing into the different brain cells. Okay, not the kidney or not the liver or not the bone marrow cells. Okay, so that is why adult stem cells are somehow also called as a paradigmatic stem cells. They have the ability to now produce the tissue specific types of the cells. Now we will consider an example of the stem cell which is present in the bone marrow, and these stem cells are called as hematopoietic stem cells, and we can write them as HSC, hematopoietic stem cells. These are also called as a paradigmatic stem cells. And why they are called as a paradigmatic stem cells? Because these cells have now ability to produce the specific cell lineages which are related to a particular type of the tissue, and how it is going to happen we will see in just a moment. Okay, so one of the most important characteristic feature of the stem cells is that wherever they are present in your body, they are always present in the limited number. Why? Because I have already told you that the stem cells have the ability to self renew. They have the ability to regenerate themselves. They can reproduce, and they can produce their own kinds, their own type of the daughter cells, so they can repopulate themselves. But they have the specific lifespan, and after that lifespan they undergo the process called as apoptosis, that is a programmed cell death. And that's why their number is basically kept in the constant quantity. And similarly, majority of these stem cells which are produced when there is a need in the body, they are undergoing the differentiation process and differentiate into the variety of other cells, but all these cells are related to a particular tissue, as I've already mentioned. Okay, so that is very important. So although they have the cell revenue capacity, although they have the regeneration ability, but the number is kept in constant by the apoptosis process and by their differentiation into the variety of the other cell lineages. Similarly, the age is very important factor for determining the number of the stem cells which are present in your body. Because when an organism age, not only the human beings but all the other types of the organism, when they are undergoing the aging process, the self renewing ability of the stem cells are decreasing. That is very important. Okay, so self renewing ability of the stem cells is decreasing with the age. That is, their ability to repopulate themselves is also tremendously decreasing. And definitely when the repopulating ability is decreasing, their ability to differentiate into the other cell lineages specific to a tissue are also decreasing. Okay, so generally it affects the entire body. As you already know, whenever the aging process occurs, the different types of the cells production or the self renewing abilities decreases. So same happens with the stem cells. Now there are two scenarios which are faced by a stem cell in its lifetime. Okay, and the number one is the when there is no immediate infection, no insult to the body occurring at a particular time, and the body is at the calm and constant and stable level. Okay, so in this particular condition, what will stem cell will do? Okay, stem cell will remain in the quiescent state. You can say in the silent state. And what is that quiescent or silent state? In this state, it will remain silent, cool, and calm, and it will self renew. Self renew mean it will regenerate or reproduce, and it will produce those daughter cells which have the characteristic features of the stem cells. Mean all the daughter cells which are produced from any particular stem cell, all these daughter cells still have the ability to self renew, to regenerate and repopulate their kind. And similarly, they also have the ability that whenever the time will come, they have the ability to differentiate into the other cell lineages as well. Okay, so this is what happening when there is no immediate infection or no immediate threat to the body and no bacterial viral infection within the body. Okay, now the second scenario is that whenever there is actually the immediate infection in the body or there is an insult to the body, and it is also seen that even after the chemotherapy in the cancer patients, when majority of the healthy blood cells are also undergoing the dying process along with that of the infectious cells. So in that particular case, the hematopoietic stem cells and all the other stem cells of the body basically have the great self-renewing ability. Mean they are greatly repopulating themselves. They are not only repopulating themselves, but they are also differentiating into the other type of the lineages, other cell lineages, which are actually diminished by the chemotherapy process or during an infection or any insult which is taking place in the body, like any bacterial or the viral or parasitic infection that is taking place in the body, and it is basically destroying the normal cells of the body. Definitely these normal cells of the body should be repopulated, and this is entirely done by the stem cells. So they not only self renew, but they also differentiate and produce those cells which have undergo the death process by these types of the infections or the chemotherapy procedures. Okay, so these are the two scenarios. Now we will see a diagram, and in this diagram we will be looking into detail that what are actually, I have already told you that in this lecture we will be particularly discussing the hematopoietic stem cells as an example, because it is directly related to the immune cells and generally the immune system. Okay, so what are actually the hematopoietic stem cells? First of all, the hematopoietic stem cells are the stem cells that are present in the bone marrow. Okay, so these are the adult stem cells, and these stem cells basically have the ability to give rise to the cell lineages which are related to a particular tissue. Okay, so I have already told you that the hematopoietic stem cells, just like the other cells, basically have one of the most important feature, that is the self-renewing ability. Okay, it can regenerate and repopulate its own kind and produce the daughter cells which have the same characteristic and feature just like that of the parent cell. And the second important thing, and this is, I have already told you that this is going to take place when there is no immediate infection to the body. Okay, but what happens when there is an immediate infection or immediate threat to the body, and there is any viral or bacterial infection that is undergoing the process? Okay, now in that condition, that hematopoietic stem cell will undergo lineage commitment. And what is lineage commitment? Mean that hematopoietic stem cell will decide that now I have to produce the specific cell lineages which are related to the immune system. That is why it will decide that I have to produce the certain type of the immune cells which are required by the body at this time, at this crucial time of the infection. Okay, and one thing you have to keep in mind, that once it undergoes the lineage commitment process, mean it has decided that now I have to lose my own identity and I have to undergo the division process and differentiation process, and I have to produce the different types of the immune cell lineages so that the body can effectively fight with the infection. Once it is decided by the hematopoietic stem cell, its ability to self renew will be diminished, will be stopped. Okay, so it can no longer self renew. Okay, so it cannot regenerate its own kind. now it would be another type of the cell. Okay, so now whenever the hematopoietic stem cells which are present in your bone marrow, they have decided to undergo the lineage commitment, it can choose for either of the two lineages. Okay, it can undergo either the myeloid lineage commitment, this one, or it can undergo the lymphoid cell lineage commitment. The meaning of this thing is that either this cell is going to produce the myeloid cells or it is going to produce the lymphoid cells. Okay, what are myeloid and what are lymphoid cells and what are these two families, we are going to discuss in just a moment within this diagram. Okay, so first of all, if that hematopoietic stem cell basically have decided to undergo the myeloid lineage commitment, so that stem cell is going to produce a progenitor cell. Okay, that is also a kind of the daughter cell, but now this progenitor cell, which is now called as a myeloid progenitor cell, they have lost its self-renewing ability, so it can no longer self-renew. First thing. Second, this myeloid progenitor cell now will undergo the further differentiation into the variety of the cells of the immune system. And what are those cells? First of all, this myeloid progenitor cell will differentiate and produce the three other important progenitor cells, that is the granulocyte monocyte progenitor cell. Okay, it will produce eosinophil progenitor cell and it will also produce basophil progenitor cell. Meanwhile, it will also produce the megakaryocyte progenitor cell and the erythroid progenitor cell. Okay, so these are almost 5 different categories of the progenitor cells which will be produced from the main myeloid progenitor cell. Okay, so you have to just follow the diagram. Now this granulocyte monocyte progenitor cell basically have the ability to differentiate into the two types of the cells. Okay, the one is the neutrophils, and you all know neutrophils are one of the type of the blood cells, and you can say these are one of the abundant type of the blood cells, which are basically immune cells which are present in our blood, and they play a very important role as a first line of the defense against the bacterial infection by phagocytosing them, and they release a certain type of the antimicrobial substances against these pathogens as well. Okay, so and neutrophils are very important, make the dense population of the immune cells, and they provide the protection against the bacterial infection. I will just talk briefly about the functions of these immune cells, because we will have a separate video on the detailed function and differences of these immune cells. So now I have told you that the granulocyte monocyte progenitor cell will either differentiate into the neutrophils or will undergo the differentiation into the monocytes. Okay, now what are the monocytes? Monocytes are the specialized type of the immune cells which are usually present in the blood, and whenever they are circulating in the blood, they are called as a monocytes. But when this blood takes them into the tissues, these monocytes will differentiate either into the macrophages or into the dendritic cells, and both macrophages and dendritic cells are the type of the phagocytic cells. Okay, so both undergo the process of the phagocytosis, and in which they basically engulf the foreign antigens, process them, and present them to the variety of the cells of the adaptive immune system, and one of those cells are the T cells. Okay, so they present the pathogens or the antigens to the T cells and mediate the adaptive immune responses. Okay, so just a brief look into the functions, but the most important thing is that to which particular cell types they are undergoing the differentiation. Okay, so I'm going to repeat it, that myeloid progenitor cell will basically give rise to the 5 other progenitor cells. Number 1, granulocyte monocyte progenitor. Number 2, eosinophil progenitor. Number 3, basophil progenitor, megakaryocyte progenitor, and the erythrocyte progenitor cell. Okay, and all of these progenitor cells now will give rise to their own different types of the cells, such as a granulocyte monocyte progenitor will give rise to the neutrophils as well as monocytes. Similarly, monocytes, when get into the tissues, they differentiated into the macrophages and dendritic cells, performing the specific functions. Similarly, the eosinophil progenitor cell will be differentiated into the eosinophils, and eosinophils are one of the most important type of the white blood cells, and they have the ability to combat the parasitic infections. Whenever there is the parasite entry into the body, the most important cells of the immune systems which are highly active are the eosinophils, okay? And they also produce various types of the allergic reactions, and during these reactions they release various types of the toxic proteins, and just by releasing these toxic proteins they basically mediate the inflammation process. The third category of the progenitor cell, which are the basophil progenitor cells, they are going to give rise to the basophils, and basophils, just like the eosinophils, play a very important role in mediating the variety of the allergic responses and producing the inflammatory responses in the body. And they produce one of the most important chemical, which is called as a histamine, and the other types of the chemicals which can mediate the inflammatory responses in the body. And these inflammatory responses and allergic reactions are very important during the infection process, because they are going to inform the other cells of the immune system which are present far off the places, to inform them that there is certain type of the infection that is going to take place in that particular red zone or reddened area, and you have to quickly move into this area to combat with the viruses or the bacteria or any type of the infection that is actively going on in this place, okay? So is basophils and eosinophils have somehow related functions, because both of them are very important in mediating the allergic and inflammatory responses, and both of them fight against the variety of the parasites, okay? Another type of the cell that is going to be directly produced from the myeloid progenitor cell, that is the mast cells, and we will be discussing their functions in detail and the characteristic properties in the coming lecture. Similarly, the fourth type of the progenitor cell which is going to produce from the myeloid progenitor cell is a megakaryocyte. And megakaryocytes are actually the large cells which, when undergo the fragmentation, they produce the smaller cell types which are called as a platelets. They don't have the actual characteristics of the cells, but we call them as cells because they play a very important role in the blood clotting process. And what are their characteristic features and what is the structure of their cell, we will be discussing them when we will have a detailed lecture on the properties of the immune cells and the functions, okay? So the fifth category of the progenitor cell, which is very important in this lineage, is the erythroid progenitor cell. And the erythroid progenitor cell are going to produce one of the abundant cells of your body and very important cells of your body, that is the red blood cells or the erythrocytes. And the function of the red blood cells, you all know, is to carry the oxygen and the carbon dioxide from the different parts of your body and just providing them with the right amount of the oxygen. Okay, now if the cell that is the hematopoietic stem cell basically undergo another decision, and it basically decides to undergo the lineage commitment for the lymphoid family of the cells, so now it will undergo the differentiation into the lymphoid progenitor cell. And now the lymphoid progenitor cell have the ability to differentiate into the nature killer cells. And the nature killer cells, as you all know, these are the types of the lymphocytes, and these are very important for the innate immune responses. And these cells have the ability to identify any type of the cell which is under the stress, any type of your body cell which is under the stress, whether it's a tumor cell, virally infected cell, parasite or bacterial infected cell. All these cells can be easily recognized by the nature killer cells, and they don't require any help from the other type of the immune cells, because nature killer cells have the ability to identify the certain cell surface markers over the surface of the cells of your body. And usually when the tumor is occurring or when there is virus or bacterial infection, those specific markers from the surface of your normal body cells have been lost, okay? So nature killer cells have the ability to identify that now these cells don't have those markers which were once the characteristics of the normal cells. So these are not our normal body cells, these are now the viral or bacterial infected cells, okay? So now I have to kill them actively. So it doesn't require any type of the prior activation from the other cells of the, you can say, adaptive immune cells or the innate immune cells. They have their own recognition for the cell surface markers, and whenever they are absent from the surface of the particular cells, they consider them as foreign cells, although they are cells of your body, but they are virally infected, okay? So it will basically destroy them. So now the second thing with the lymphoid progenitor cell is going to produce are the two important type of the progenitor cells, that is the daughter progenitor cell, that is a T cell progenitor cell and the B cell progenitor cell. T cell progenitor cell is going to differentiate further and in the thymus, and you all know this process is going to take place in the thymus. And if you don't know, I have a separate video on this lecture as well, on this topic as well, that how the maturation and differentiation of the T cells is going to take place in the thymus, where I have discussed in detail all the steps which are basically required for this kind of the process or for this differentiation. So I'm just going to have a brief look here that the T cell progenitor cells are going to differentiate and produce the T helper cells and the cytotoxic T cells when they come out of the thymus. Okay, and the function of the T helper cells is to undergo the antigen presentation by the antigen presenting cells and going to basically activate the other type of the immune cells such as macrophages, dendritic cells, natural killer cells, and also cause the activation and production of the more and more effector T cells, just boost up the immune response. Okay, so they don't have the direct impact on the virus or bacteria or the parasitic infected cells, but they activate the other type of the immune cells, and play a very important role, and they act as a bridge between the adaptive and the innate immune responses by activating their cells. Similarly, the cytotoxic T cells, as their name indicates, they have the direct impact on the cells which are infected with the viruses or the bacteria or the other types of the parasites. And whenever they encounter such type of the cells, they are going to release certain type of the chemicals. And those chemicals are the granzymes and perforins. And they are very important for causing the apoptotic cell death of these particular type of the infected cells. Similarly, the other type of the progenitor cell which are going to produce from the lymphoid lineage are the B cell progenitors, and they are going to produce the B cells, and we all know B cells produce antigen specific type of the antibodies, and these antibodies are very important for marking the particular parasite, the bacteria, so that it can be easily recognized by the other type of the immune cells and the direct immune responses can take place. Similarly, the dendritic cells are also produced directly from this lineage. So you can see that the dendritic cells are the type of the cells which are produced in both the lineages. Okay, that is the myeloid lineage and the lymphoid lineage. Now, what are basically the genetics behind the this hematopoiesis process or this lineage commitment process? Because definitely if a cell is going to decide that it is undergoing the specific lineage commitment, that is whether the myeloid lineage commitment or the lymphoid lineage commitment, definitely there are certain decisions which are going to take place at the genetic level, because all the activities of the cells, their self-renewing ability, their lineage commitment abilities, all these characteristics are controlled at the genetic level, that is at the DNA level. This means that there are specific type of the regulators which are working at this time in order to decide that which particular type of the lineage should be switched on or should be switched off, depending upon the need of the body, that which type of the cell lineage is required, whether the myeloid cells such as the dendritic cells, the neutrophils, mast cells, eosinophils, basophils are required by the body, or there are other type of the cells such as B cells, T cells are actually required for the body against the infection. Okay, so the most important thing here is that you have to consider this thing that let me take a color. Okay, let's take this. The cells which I am highlighting here, these are all the cells of the myeloid lineage. Okay, that is basically the granulocyte monocyte progenitor cell and all the cells related to this lineage, which are neutrophils, monocytes, macrophages, dendritic cells, and the one of the other type category of the cell, that is the natural killer cells. These are all the cells of the myeloid lineage, and they play a very important role in the innate immune responses. Okay, similarly, the cells now I'm highlighting in the this yellow color. Okay, so these are the cells of the lymphoid lineage. Okay, so these are the helper T cells, cytotoxic T cells, and B cells. Similarly, the eosinophils, basophils. Okay, these are all the cells of the lymphoid lineage. While these two category of the cells, that is the platelets and the erythrocytes. These are not the part of the lymphoid and myeloid lineages. These, because they don't play any important role in the adaptive or immune responses, they have the other characteristics. Okay, for example, platelets are involved in the blood clotting, erythrocytes in the gases transport. So they have the other functions, but I have cleared the myeloid lineage and the lymphoid lineage. Now, what are the genetics behind the commitment to these lineages is very important. And when the various steps of the investigations are still undergoing, and the various results which have been published up till now, they have shown that the most important primary genes which are important for the lineage commitment of these cells is the transcriptional regulators. Transcriptional regulators are those particular type of the proteins, other transcription factors which are very important for carrying out the process of transcription. And these are the specialized factors or specialized proteins which basically carry out the transcription of the genes that are required for the lineage commitment to either the lymphoid progenitor cells or to the myeloid progenitor cells. And the four important categories of these transcription regulators have been identified up till now, and various other primary genes are still under the investigation. And one of the most important transcription regulator in this line are the GATR2 transcription regulators, and these GATR2 transcription regulators are very important for carrying out the process of the hematopoiesis. Mean they carry out the switching of those genes. They actually switch on those genes in the DNA which are very important for the lineage commitment, whether the myeloid lineage commitment or the lymphoid lineage commitment. So whenever these particular type of the similarly, definitely when the scientists are studying these types of transcription regulators, they have studied that what are the impacts of their absence within the animal's body. And when from various certain mice these particular type of the transcription regulators have been knocked down, these animals usually die during the embryogenesis process. And that is very important, that during the embryonic life these organisms basically embryogenesis, during the embryonic life these organisms basically die because they don't have the ability to carry out the lineage commitment to produce a variety of the immune cells, and these organisms are highly immunocompromised and they immediately die during the initial stages of their life. Okay, so that GATA2 is one of the most important type of the transcription regulator which is important for the lineage commitment for these types of the hematopoietic stem cells. Another type of the transcription regulator which have been discovered now is the BMI1 transcription regulator, and it is very important for the self-renewing ability of the hematopoietic stem cells. Okay, mean it helps them to repopulate themselves, it helps them to produce the daughter cells of their own kind. And whenever the BMI1 has been knocked down from the mouse, they still can survive, but only for the 2 months. For 2 months they can survive, and then these mice die. Why? Because they don't have the self-renewing ability. These hematopoietic stem cells don't have the self-renewing ability, and when the existing stem cells basically die, when their lifespan is over, there are no new stem cells which are available for performing the particular functions and to produce a variety of the immune cells. So definitely BMI1 is one of the most important type of the transcription regulators to switching on those genes which are carrying out the self-renewing process. And whenever that particular transcription regulator is absent, the organism is going to die within the 2 months of their lifespan, because the body is not able to repopulate the white blood cells and the red blood cells. Another type of the two family of the transcription regulators which have been discovered and play a very important role in the lineage commitment are the agarose family of the transcription regulators and the notch family of the transcription regulators. Okay, so agarose family of the transcription regulators, and this family includes lots of the other types of the transcription regulators. Okay, so agarose family of the transcription regulators play a very important role for the myeloid lineage commitment. And you all know myeloid lineage commitment is going to produce the dendritic cells, the mast cells, similarly the basophil cells, eosinophil cells, neutrophils, macrophages, monocytes. it means that whenever there is an immediate infection in the body, the Ikaros family of the transcription regulator is going to be activated to produce lots and lots of the myeloid cells, because myeloid cells are the cells which provide the first line of the defense whenever any pathogen or bacteria basically gain entry into your body. So whenever there is an immediate infection, the first cells which are going to fight with them are the myeloid cells, and at that time the Ikaros family of the transcription regulators is going to switch on the gene which cause the hematopoietic stem cells to undergo the lineage commitment for the myeloid cells, so that lots and lots of these cells can be produced and can effectively combat any type of the ongoing infection in the body. Similarly, the Notch family of the transcription regulators, and among these transcription regulators, one of the most important type of the regulator which have been identified is the Notch1. Okay, so Notch1 is very important for carrying out the lineage commitment to the lymphoid progenitor cells. Okay, it causes the hematopoietic stem cell to undergo the lineage commitment towards the lymphoid cells, lymphoid progeny, and definitely it is going to produce lots and lots of the T and B cells. And these cells are very important for mediating the adaptive immune responses, because they are very important for producing the antigen specific type of the immune responses. Also, the scientists have discovered that apart from these particular type of the functions which are taking place in the body and the factors which are influencing, environment also plays a very important role on the self-renewing ability or the lineage commitment ability, which is also called as a hematopoiesis ability of the hematopoietic stem cells. And for example, when the environment is like in the active infection state, for example, your body's environment is in the active infection state, in that state more and more myeloid cells and lymphoid cells are produced. Okay, whenever your body is under the attack by the bacteria or the parasite or fungus attack or any other type of the attack, whether it's a chemotherapy is going place in your body, similarly any type of the other chemical procedure that is taking place in the body, in that alert state, the myeloid cells and lymphoid cells are actively producing. That's why the hematopoiesis process is going to be speed up. Okay, and similarly, when the environment is cool and calm in the body and there's no immediate infection in the body taking place, then the cells mainly undergo, hematopoietic stem cells mainly undergo the self-renewing process. Okay, so the environment also impact the ability of these hematopoietic stem cells to undergo either the self-renewing process or to undergo the differentiation process into the variety of the cell lineages. Okay, so in this lecture we have studied in detail the stem cells, what are their important properties, what are the pluripotent stem cells, what are actually the adult stem cells, and how these cells are going to produce the tissue specific cell types. And we have studied one of the most common example of the stem cell, that is hematopoietic stem cells, and how the age and the environment basically affect the activity of the stem cells. And we have studied the two scenarios for the working of the stem cells, that is whenever they are in the under the immediate attack or when they are in the non-attack state. Okay, also we have studied that the hematopoietic stem cells basically undergo the two lineage commitments, either into the myeloid progenitor commitment cells or the lymphoid progenitor cells. And lately we have studied the genetics behind the hematopoiesis or the lineage commitment of these cells. If you have any question, you can ask me in the comment box, and thank you so much.